Okay, welcome everybody. This is our podcast dealing with blood vessels. We're going to look at an overview of arteries and veins and talk about blood vessels, anatomy in general, and the physiology that drives circulation in order to get the blood filled with nutrients and oxygen to all the cells that need it while removing carbon dioxide and waste products in order to keep from creating a toxic environment around the cells and tissues that would kill them. Blood vessels transport blood to tissues where gases, nutrients, and wastes are exchanged and then transported back to the heart. They regulate blood flow to tissues and control blood pressure and secrete chemicals. The two circuits that carry the blood through the body are the pulmonary circuit, which transports blood between the heart and lungs, and the systemic circuit that transports blood between the heart and the body. The pulmonary and systemic circuits are composed of three different types of blood vessels: arteries, capillaries, and veins. Arteries, as we learned in the last chapter, always carry blood away from the heart, and they are the distribution system of the cardiovascular system. They branch into vessels of progressively smaller diameter that supply most of the body's tissues with blood. Capillaries are the exchange system of the vasculature. They're small diameter vessels that form branching networks called capillary beds. Here, gases and nutrients, wastes, and other molecules are quickly exchanged between tissue cells and blood through capillary walls, many of which are only a single cell thick. Veins are the collection system of the vasculature and drain blood from the capillary beds and return it to the heart. They follow the opposite pattern of arteries. Small veins merge into larger veins that become progressively bigger vessels as they progress back towards the heart. All blood vessels are tubular organs that contain a lumen surrounded by several tissue layers called tunics. The three tunics of the blood vessel wall are the tunica intima, the media, and the externa. The tunica intima is composed of endothelium, continuous with the inner lining of the heart, known as the endocardium. The endothelial cells provide a smooth surface over which blood can flow with a minimum of friction and turbulence. A thin layer of subendothelial connective tissue and a layer of elastic fibers called the internal elastic lamina are found deep to the endothelium. The elastic fibers give the vessels the property of distensibility, which is the capability of stretch when subjected to increased pressure, and elasticity, the ability to recoil back to its original size when the stretching force is removed. The middle layer of a blood vessel is called the media, and it's composed of two layers with the following features: a layer of smooth muscle cells arranged in a circular manner around the lumen, and another layer of elastic fibers called the external elastic lamina. Smooth muscle cells of the tunica media control the diameter of the blood vessel and therefore the amount of blood that flows to the organs. Smooth muscle cells of the media are innervated by sympathetic nervous system vasomotor nerves. These nerves stimulate smooth muscle cells of the media to contract in a process known as vasoconstriction, narrowing the diameter of the vessel. When sympathetic stimulation of smooth muscle cells decreases, these cells relax and the vessel's diameter increases, and this is a property known as vasodilation. The outermost tunica externa, also called the adventitia, is composed of dense, irregular, collagenous connective tissue that supports the blood vessel and prevents it from overstretching. In the thickest vessels, vasa vasorum, are tiny vessels that supply the tunica media and the tunica externa with its own blood supply. And the reason for this is that the thickness of the walls of these vessels doesn't allow it to utilize the oxygen and nutrients that are carried in the lumen. They supply oxygen and nutrients to the outer layers of the blood vessels, whose cells are too far away from the lumen to receive oxygen and nutrients by diffusion alone. Note that the structure of the blood vessel wall, which is an inner epithelial tissue lining, a layer of connective tissue, a layer of smooth muscle, and an additional layer of tough connective tissue, is a pattern that you'll see repeated in many other hollow organs, such as the gastrointestinal tract. In an example of the structure-function core principle, this pattern of organization ensures structures of hollow organs best follow their function to control the flow of substances that pass through them. The following differences are notable between the walls of a typical artery and those of a vein. Most arteries have a much thicker tunica media than do veins, and this reflects the artery's role in controlling blood pressure and blood flow to organs. In addition, the internal and external elastic lamina are much more extensive in arteries than in veins, and this reflects the fact that arteries are under much higher pressure than veins. And then one more very important feature that veins have that arteries do not are the presence of valves. This ensures the one-way flow of blood back to the heart under the low pressures that you see in the venous return. This is absent in arteries due to the fact that the high pressures generally are sufficient to push the blood from point A to point B. The thickness of each component of the arterial wall depends on the artery size and function, and is used to class arteries based on the similarity in structure and function into elastic arteries, muscular arteries, and arterioles. Elastic arteries, also known as conducting arteries, are the largest diameter arteries in the body and include the aorta and its immediate branches nearest the heart and therefore under the highest pressure of any vessels in the cardiovascular system. Muscular or distributing arteries are intermediate in diameter, contain well-developed tunica media made of primarily smooth muscle. Most of the smaller branches off the aorta are considered muscular arteries, including most of the named arteries that supply organs, such as the renal artery, supplying the kidneys. Arterioles are the smallest arteries and contain all three layers of blood vessel wall, but the layers are extremely thin and the tunica media only contains about 1 to 3 layers of smooth muscle. The smallest arterioles are called meta-arterioles, and they directly feed capillary beds in most tissue. Smooth muscle cells of the meta-arterioles are confined mostly to a circular precapillary sphincter that encircles a meta-arterial capillary junction and acts as a doorway into that capillary bed. Certain arteries also play a role in monitoring blood pressure and detecting the concentration of chemicals in the blood. Pressure receptors, known as baroreceptors, are found in the aorta as well as the common carotid artery and are located in the neck. They're also found in the carotid artery and aorta, another group of receptors known as chemoreceptors that are able to detect blood oxygen and carbon dioxide levels as well as the pH of the blood. Veins typically outnumber arteries and their lumens have a larger average diameter. Veins have the following characteristics. 70% of the total body blood is located in veins at any given moment. That's why they're called capacitance vessels. These include both the systemic and the pulmonary veins. This allows the veins to function as blood reservoirs because the blood can be diverted from veins to other parts of the system. Veins typically have much thinner walls, fewer elastic fibers, less smooth muscle, and larger lumens than arteries, in addition to the presence of valves. Veins are also classified by size. Venules are the smallest veins and drain blood from the capillary beds. Tiny postcapillary venules consist of little more than endothelia and surrounding connective tissue. The structure enables them to exchange material with the surrounding interstitial fluid. The three tunics become more distinct as the venules merge to become larger venules and then veins. Most veins have a thin tunica media with a few smooth muscle cells, and the diameter only changes slightly as they vasodilate and constrict. Many veins contain venous valves, which are extensions of the intima that overlap and prevent blood from flowing backward in the venous circuit. This is especially numerous in the veins that are located below the heart, where the blood flow towards the heart is opposed by gravity. And you can see here a chart showing you the different types of arteries and veins, their diameters, their structures, and their functions. This is table 18.1 in your text. Now, atherosclerosis is the leading cause of death in the developed world. Arterial disease that affects large and medium-sized muscular arteries is frequently seen amongst Western populations due primarily to the presence of the high amount of saturated fat and cholesterol present in our diet. The disease is characterized by the formation of arteriosclerotic plaques, which are buildups of lipids, cholesterol, calcium salts, and cellular debris within the arterial tunica intima. The plaques tend to form in regions where blood undergoes sudden changes in velocity and direction of flow, such as the branching points or where vessels curve. Current evidence indicates that plaques are generated in response to some sort of injury to the endothelium by high blood pressure, certain types of cholesterol, previous infections, toxins in cigarette smoke, a high and prolonged blood glucose level, and a blood concentration of a chemical known as homocysteine. The vessel wall gets inflamed and attracts phagocytes that attempt to clean up the area, and this actually results in damaging the vessel, and eventually the damaged area turns into a plaque. The plaque is located in the intima, but changes also occur in the media. Smooth muscle cells proliferate and secrete an extracellular matrix with protein fibers and ground substance that surrounds the plaque. If the plaque is exposed to blood, a clot can form in a vessel and obstruct blood flow entirely, as occurs in a myocardial infarction, which would be a blockage in the heart, or a stroke, which would be a blockage in the brain. An estimated 10% of the world population has atherosclerosis, but its true prevalence is difficult to determine because most patients don't display symptoms in the early stages. Much of the treatment of atherosclerosis focuses on reducing factors that injure the endothelium. This includes dietary modifications, a change in physical activity level, agents to lower cholesterol, control of blood glucose, cessation of smoking, and management of high blood pressure, and in severe disease, surgery or invasive procedures that may be necessary to open or bypass the blocked vessel. Now, you might wonder, okay, why is it that when the blood vessel narrows, the likelihood of a clot is inferred? And the reason is that the rate of flow through the restricted area, through the narrowed lumen, is slower. And one of the things we're going to find out about blood is that as blood flow slows down, blood tends to clot. Okay, so that clot, if it's traveling, is referred to as an embolus. If it's lodged in a blood vessel, it's called a thrombus, and of course that's going to cut off blood flow to downstream tissues and cause them to infarct and die. Now, eventually, any embolus is going to jam at some point in a capillary bed, provided it persists in the circulation long enough, because of the fact that the smallest vessels, the capillaries, are so narrow that red blood cells often have to go through single file like a stack of pennies in order to get from one side of the capillary bed to the other. A embolus certainly would jam in a vessel with such a narrow diameter. Vascular anastomoses are locations where vessels connect through pathways called collaterals. Arterial anastomoses exist in many organs such as the heart and brain, as well as around joints. New arterial anastomoses can be formed when blood flow through the artery is insufficient to meet the tissue's metabolic needs. Venous anastomoses are the most common type. Here, neighboring veins are connected by small collaterals. Smaller veins are often so interconnected that they form a web-like pattern. Arteriovenous anastomoses are where an artery empties directly into a vein without passing through a capillary bed. Now, hemodynamics examines the physiology of blood flow in the cardiovascular system. We have to be aware of the fact that gradients drive flow. Flow always is from high to low pressure, whether it be a gas or a liquid. Other types of gradients that exist include concentration gradients and electrical gradients. The heart provides the force that drives the blood through the blood vessels by creating a pressure gradient, which is an example of the gradient core principle. Pressure is highest near the heart and decreases as blood moves further away. Blood flows down its pressure gradient from high to low pressure in the peripheral vasculature. Blood pressure is simply the outward force that the blood exerts on the walls of the vessels. It's expressed in units of millimeters of mercury, or mmHg, which is the force exerted by a column of mercury 1 millimeter in height. We'll get into where that comes from in a minute. It varies dramatically in different parts of the vasculature. It's highest in the large systemic arteries and lowest in the large systemic veins. So now let's talk a minute about why millimeters of mercury is a unit of pressure. And this goes to a device that's called a barometer. Okay, now a barometer is a U-tube. It's closed at one end and open at the other, and is filled with a liquid called mercury, which is one of two metals that's a liquid at room temperature. Happens to have a meniscus that is convex instead of concave, unusual for a liquid. So that's our mercury in our barometer tube. And the weight of the air on the mercury is the atmospheric pressure, and then we measure with a ruler that has millimeter marks on it the number of millimeters of mercury that we see in the part of the tube that is closed to the atmosphere. Okay, and that tells us something about the atmospheric pressure. So that's where that terminology comes from. The magnitude of blood pressure in a gradient is the main factor that determines blood flow, or the volume of blood that flows per minute. Generally, blood flow matches cardiac output, averaging 5 to 6 liters a minute. Blood flow is directly proportional to a pressure gradient, meaning that blood flow increases when pressure gradients increase, and vice versa. Another factor that determines the blood flow is the resistance, and it's defined as any impedance to blood flow. Blood flow is inversely proportional to resistance, that is, as resistance increases, flow will drop. The velocity with which the blood flows is determined by the cross-sectional area of the vessel. As the arterial system branches into smaller vessels, the total cross-sectional area goes up. This increase in area causes the velocity of blood flow to decrease. For this reason, the velocity of blood flow is the fastest in the aorta and slowest in the capillaries. Area is simply a mathematical calculation of the area of a circle made by a cross-section through that circle. It's equal to pi times the radius of the circle squared, as shown here. The radius of an arteriole or a capillary is small, so the cross-sectional area of such a vessel was also very small. However, these vessels are more numerous than the large vessels with a large cross-sectional area. Therefore, the total cross-sectional area of a small vessel is much greater than that of a large vessel. As an example of how this works, take a large vessel with a cross-sectional area of 5 square centimeters, and each of 5 smaller vessels with an area of 2 square centimeters. The total cross-sectional area of the small vessels, 10 square centimeters, is double that of the large vessel. So it's easy to see why cross-sectional area increases as arteries branch into millions of smaller vessels. Velocity decreases as cross-sectional area goes up because the same volume of blood is now filling the equivalent of a much larger container. Blood is spread out over a wider area as the vessels branch, causing the flow to slow. And this is important because a slower velocity of blood flow allows efficient gas and nutrient exchange in the capillaries, and it also provides enough time to remove the waste products, the carbon dioxide, the ammonia, the urea, all that other stuff that the cells make as they metabolize. 3 main factors that influence blood pressure are the resistance, the cardiac output, and the blood volume. The peripheral resistance is any factor that hinders blood flow through the vasculature, contributing to the overall resistance of the circuit. Vessels near the heart contribute little to the overall resistance, while resistance is greatest further from the heart in the body's periphery. So, as peripheral resistance goes up, blood pressure also goes up. Resistance is determined Also, by the blood vessel radius. The resistance varies inversely with the blood vessel radius. As the radius increases, the resistance to blood flow drops, and vice versa. Blood viscosity is another factor. The viscosity is defined as the inherent resistance that all liquids have to flow. The more viscous a liquid, the more its molecules resist being put into motion and staying in motion. Blood has a relatively high viscosity due to the number of proteins and cells it has. And the length of the blood vessel. Basically, the longer the blood vessel, the greater the resistance. The more pressure is needed to propel the blood through a long vessel than a short one. This is one reason why resistance in the pulmonary circuit is lower than in the systemic circuit. And it's also why blood pressure tends to go up in individuals who are obese, because when you put on bad weight, adipose tissue, your blood vessels don't get more numerous, they get longer. And so that increases the afterload that the heart has to pump against. The cardiac output is simply the product of the stroke volume and heart rate. Cardiac output and peripheral resistance are two factors that determine the pressure gradient that drives circulation. The relationship is expressed as follows. The difference in pressures between two parts of the circulatory system is equal to the product of the cardiac output and the peripheral resistance. The pressure change is caused by altering the cardiac output or the peripheral resistance. When the cardiac output increases, the blood pressure goes up, and vice versa. And, as you probably guessed, when the peripheral resistance goes up, you're going to see a greater difference in the pressure difference between 2 points in the circulatory system. The final factor that determines overall blood pressure is blood volume. The total volume of blood is directly linked to the amount of water the blood contains. When blood contains more water, the blood volume goes up. As blood volume increases, blood pressure goes up, and vice versa. Small vessels in the blood volume, small increases in blood volume are offset by vessels that stretch, which is a property known as compliance. Veins are the most compliant vessels, and they stretch to adjust added fluid when blood volume increases with a rise in pressure. Blood pressure remains fairly low throughout the pulmonary circuit. However, blood pressure changes significantly as blood travels through the systemic. The blood pressures in the two circuits are dramatically different, averaging 15 millimeters of mercury in the pulmonary circuit and 95 in the systemic. Systemic arterial pressure is something that we can examine. The pressure profile of the entire systemic circuit is highest in the aorta and the elastic arteries and declines slightly as it spreads throughout the muscular arteries. The mean arterial pressure is the average pressure in the systemic arteries during an entire cardiac cycle, and it's about 95 millimeters of mercury. The heart has both contraction and relaxation periods. So pressure gradients generated by the heart pulsate. They rise during ventricular systole and decline during diastole, leading to two separate pressures in the arteries. The systolic pressure averages about 120 millimeters of mercury, while the diastolic about 80 at rest. The difference between the systolic and diastolic, which is 40 millimeters of mercury, is the pulse pressure. Arterial blood is usually measured in the arm with an instrument called a sphygmomanometer and a stethoscope. The mean arterial pressure is difficult to measure directly. It's approximately equal to the diastolic pressure plus 1/3 of the pulse pressure. So we can see the equation here. The pulse pressure, remember, is equal to the systolic pressure minus the diastolic pressure. So keep these two equations in mind. I guarantee you're going to see them again on the exam and in lab. Okay, so don't forget these two equations. Here. Pressure doesn't change much in the pulmonary circuit, remaining fairly low from the pulmonary artery to the veins. However, pressure does change significantly as blood travels through the systemic circuit. Systemic capillary pressure is the pressure at the arterial end of a capillary bed, which is about the same as in the small arterioles, about 35 millimeters of mercury. But at the venular end of the bed, pressure is dropped to 15 millimeters of mercury. This pressure decrease is due to the reduction in blood volume that takes place in the capillaries as they lose fluid to the surrounding tissues. And what you're looking at here is just a diagram showing you the difference in overall blood pressure, as well as differences between systolic and diastolic pressure as we move away from the heart. Okay, so you can see here the average pressure, the mean arterial pressure in the black trace. And as we get away from the heart, a couple of things happen. We're further away from the pump, which means the influence of the pump is going to be minimized, and also we're experiencing frictional resistance as the blood is rubbing against the walls of the vessels, and the vessels are getting progressively smaller. Okay, so the diameters of the vessels are dropping, and you can see that the most dramatic drop is from the arterioles to the end of the capillary beds. And once we get to the end of the capillary beds, the pressure is quite negligible. Okay, and then what happens is we enter the venules, the veins, and the vena cava, is that the pressure continues to drop. It increases just a tiny bit as we get back towards the return on the heart, and that's due to coalescence of the blood, but also the drawing action of the heart along with the action of the skeletal muscle pump and the pulmonary pump. You can also see how the difference in the systolic and diastolic pressures decreases the further away we get from the heart. Now, important point here is that when you take a pulse, you always want to measure in a superficial artery. Okay, place like the radial artery, the carotid artery, arteries in the inguinal region, in the popliteal region on the top of the foot are all places where arteries run close to the surface, and there's not a lot of overlying tissue to confuse or conflate your readings. And so these are typical places where you would take a pulse. You can't take a pulse in a vein because the pressures are too low to detect. Pressure declines even further in venules and veins, dropping to 4 millimeters of mercury in the inferior vena cava and as low as 0 in the right atrium. The low pressure is largely due to the high compliance of the veins and the declining resistance as the vessels merge and get larger. The venous blood has to be returned to the heart at the same rate that it's pumped into the arteries. The venous circuit is under such low pressure that there isn't much of a driving force to propel venous blood back to the heart, so valves prevent the backflow in some veins, specifically those below the heart. Smooth muscle in the vein walls can contract under sympathetic stimulation and increase venous return. And the skeletal muscle pump can squeeze the blood through these valves and work it back up to its heart. And that happens when you move. You walk, you sit, you stand, you jump, you run. You can see the action of the skeletal muscle helps to aid in the venous return. More blood goes into the heart, more blood comes out of the heart. The result is that you have a greater cardiac output, and that's one of the benefits of exercise.